Hi guys, my name is Imad. Today I'll be talking to you guys a little bit about the procedure to run a shear stress model. So shear stress is a technique to used to study the effect of drugs uh, in in a physiological condition where blood, for example, endothelial cells in your blood vessels where blood is constantly moving. Today I'm running an experiment on endothelial cells, and I'll walk you through what I do for this process. For the setup itself, I use two different types of tubings. First, I use the hard tubing by Masterflex, the hard tubing, and then the soft tubing for the interior part to connect to the slides. I use ibidi slides, which are 1.8 lure uncoated. Then I have the masterflex pump, and then an incubator, a pulse dampener, tubing, and I'll walk you through the process. So initially, we want hard tubing from the masterflex pump itself, which is going to be running from the back of the incubator in between the jaws through a back opening into the incubator itself. Here I'm using an homemade collection tube basically, or 50 ml Falcon tube as a reservoir for my media. I have one of the tubings coming into the reservoir, sucking up the media from the hard tubing, and then hard tubing connecting to the pulse dampener. From the pulse dampener, I have hard tubing and then soft tubing connected. So I'll be running slides in series, so that's why I have two joints right here and right there, and then a return tube into the reservoir itself. I also have a gas exchange pipe on the top with the filter on. I have the media running initially, so it's recommended to have your media running through the tube just to warm up the media and the tubing just to avoid air bubbles. Initially, when you're first running this experiment, what you should do is initially where you're getting, you should count the number of drops in a 50 ml Falcon tube or 15 ml Falcon tube for about 2 minutes. The first minute I would discard the number of drops, and then for the second minute I would count the number of drops. So this will help you determine the flow in the tubing itself. The flow varies, depends on the tubing that you're using, the length especially, and if it's hard tubing or soft tubing. I recommend using hard tubing in the back through the pulse masterflex pump itself, as using soft tubing right here in between the pump would reduce the life of the soft tubing. After you have that all set up, you're gonna go to calculate the flow itself. To calculate the flow, I'm gonna go here. Since I'm gonna be using 1.8 slides, I go down here, I calculate the mLs per minute, and usually I run low shear at between 10 and 15 dynes, and then high shear at 20 dynes. So initially when I counted the flow rate to be about 45 ml drops or ml per minute total, and then the approximate dyne would be 10 dynes. So I adjust my tubing. You would use the formula that's provided up in the document to calculate the shear itself, which would be right here. this would be your shear rate or velocity, dynamic velocity, and the shear stress, T is the shear stress, N is the dynamic velocity, and Y is the rate itself. So based on the calculations, you're gonna go through that. Because these plates were uncoated, I coat these plates with a fibronectin coating and let the slides sit in the incubator for overnight. I let the slides fibronectin sit in the slides overnight, and then the next day I plate the cells. I wash the cells and then start the shear stress itself. I'm going to show you initial shear of the cells itself. With no shear, the cells tend to look without any shape. And I usually run the shear depending on your experiment itself. You could run the shear itself for different periods of time at different shear rates. And then after my shear runs, you tend to see elongation of the cells in the direction of the flow itself. Alright, now I have the slides connected to the soft tubing directly to the slides itself. I find that better than using any adapters or connectors directly to the tubing. This helps with the flow, not restricting the flow within the tubing or the slides itself. Just as a recap, I use hard tubing from the diastolic pump or masterflex pump. The one part of the hard tubing is going to be connected to the reservoir, taking the media in. The other part to the diastolic pump to collect the media. Then the other part of the pump of the diastolic pump is going to connect to soft tubing, connecting to one of the slides, and then the slide soft tubing to, for my case I'm using 2 slides, and then the soft tubing directly into the reservoir itself, media reservoir. In addition to this, just make sure you avoid air bubbles within the tubing as it causes cell detachment. Have your plates coated with fibronectin. It prevents cell detachment with the shear itself. Also gives better results. Just keep an eye for contamination. As the tubing goes in and out, it's easy to get contaminated. Just keep an eye for contamination, the smell, or change in media color. And then after your designated time for shear, I use these Kelly clamps to clamp off the tubing at one end of the tubing before I remove it, just to prevent any media from leaking out or seeping out, because there's still going to be media within the tubing even after you stop or pause the pump itself. I use the clamps to clamp and then connect to the slides itself. It helps. Also, I use a rubber mat just to prevent any contamination or media to fall down into the bottom trays. And after you're done with your shear, you're going to take images and quantify it immediately. If you're doing RNA extraction or anything like that, immediately within 15 to 30 minutes should be your window to do any of those. Thank you, and hopefully this video was informative.